Thank you all for joining us for the first webinar in the Advancing Care Through Genomics virtual speaker series. This is a part of the Individualized Medicine virtual experience. And if you enjoyed this experience, we encourage you to stay in touch with us through our website. Look forward for other webinars that we will be offering over the next several months. I'm super excited to be introducing Dr. Russell today. He will be talking with us about viruses for cancer therapy, a promising treatment in need of a personalized approach. Mayo was lucky to recruit him in 1998. He's established the Department of Molecular Medicine and really was the first to orchestrate the first in human testing of oncolytic measles and vesicular stomatitis viruses. His introduction is long, but to make a point, we have over 130 registrants for this webinar in less than 2 weeks. So without further ado, I'd like to introduce Dr. Russell to share with us his knowledge and expertise.
Okay, thanks very much, Myra. Appreciate that introduction. And let's get going. Viruses for cancer therapy. And the format for this this series of lectures is actually quite interesting. So I was asked to give a fairly short presentation, 10 or 15 minutes, and then to take Q&A. So this is not a long lecture. It's more of an introduction to the topic, and then we'll take it from there. So can you advance the slide for me, please? I don't think I have control of that, or do I? Oh, I do. Okay, so I have a financial conflict of interest to disclose. So I'm a co-founder of Virotherapy, which is a company spun out of Mayo Clinic to develop oncolytic virotherapy commercially. And so Mayo and I do have a commercial conflict here. Next slide, please. Next, can I have the next slide, please? It's not advancing on my screen. Is there a problem your end?
I am working on it right now. I apologize. All right. Are you able to see it here?
Yeah. Okay, so viruses are, and I'm sure you're all familiar with this, nature's smallest organisms are much smaller than bacteria. And essentially a virus has a protein shell containing just a few genes, which may be DNA or RNA. The shell injects into its genes into a cell, turns it into a virus-producing factory, and the viruses, the progeny viruses, then spread. And viruses cause many illnesses: polio, measles, mumps, chickenpox, etc. And more recently, COVID-19 is a virus that we're all painfully familiar with. Next slide. So viral life cycle we needn't go into in detail, but the virus, having injected its genome into a cell, then makes more copies of itself. So the genome is amplified in the cell. The proteins encoded by the viral genome are made in the cell, and new progeny particles are put together. But the bottom line is that for a typical virus, one virus gets in, and then maybe up to 10,000 progeny viruses are released a few days later when the cell dies. Next. So there are a lot of viruses out there. There are more than 4,000 viruses in nature known to infect various different species of mammal. And you can see them laid out here. And they come in all different shapes and sizes, but they're all very small. So they're sort of nature's nanoparticles. And they may have RNA or DNA inside them, and it may be single-stranded, double-stranded, circular, linear. And then it's wrapped up in protein, and it may be icosahedral symmetry or it may be helical symmetry. And then there may or may not be a lipid envelope around that protein shell, in the surface of which is some additional proteins to help the virus get into its target cells. So you got all these different choices of viruses, and you know, we generally view viruses as being a scourge because of all those diseases that they cause. But they're actually turning into a great untapped bioresource. And, you know, an awful lot of what's going on in medicine now depends on viruses. So gene therapy uses viruses to deliver genes. Regenerative medicine, an awful lot of what's going on there requires gene transfer into cells that are being used for therapy. And then virotherapy, which we're talking about today, where viruses are being used to attack cancer. The next slide is just a schema of what happens during oncolytic virotherapy. So a virus is administered to a patient with the sole purpose of impacting their cancer. And it's a two-step therapy. So in the first step, the virus infects the tumor and spreads within the tumor, causing a trail of destruction, which is a sort of inflammatory killing with a lot of danger signals going on in the tumor. And that draws in the anti-tumor T cells, which are then amplified. And so the two stages to oncolytic virotherapy are first tumor destruction, and then second, long-term disease control because of the activation of the immune system. Next slide. So we've been developing oncolytic viruses at Mayo Clinic for quite some time. I was recruited to Mayo back in 1998 to start the molecular medicine program. And when we got that program going, our sole purpose was to build this translational machine to allow us to take viruses all the way through from design in the lab through to clinical testing. And of course, for that, we needed quite a lot of translational infrastructure in place, not just to build out clinical protocols, but to do the vector manufacturing, because you can't just take stuff you've made in the lab and use it clinically. You need this GMP manufacturing capability. And we needed then to be able to run whatever tox farm studies FDA would need in order to support clinical implementation. So, you know, we have those capabilities at Mayo, the manufacturing run by Dr. Federspiel, the farm tox by Dr. Kawai Peng. And then the study I'm going to talk about first is a study where we took a measles virus that we'd engineered for cancer therapy, and we gave it intravenously to patients with multiple myeloma. Multiple myeloma is an incurable blood cancer in which antibody-producing B cells are transformed, and they live in the bone marrow, and they cause multiple solid tumors throughout the bone marrow. And of course, this disease is ultimately fatal. So we had a trial in which we started with a very low dose of measles given intravenously, single dose, and we went all the way up to 10 to the 11th, which is an enormous amount of measles virus. So we saw very little activity at the lower dose levels in this trial, and we were sort of worried about whether we were ever going to see anything. But then once we got to the top dose level, it got interesting. And can we see the next slide, please? So we had this star patient, Stacy Erholtz, who was very much in the media when she first responded to the virus. And you can see this Star Tribune article sort of went viral, no pun intended. Because Stacy had a complete response to virotherapy. And I'll just walk you through that because it is important. Next slide. So she was only 49 years old. She had a particularly unpleasant type of myeloma that had been, it had become refractory to all available myeloma treatments. Stacy had had 2 stem cell transplants and was relapsing quite rapidly after her second stem cell transplant. She came to the clinic with multiple solid tumors visible on her PET CT scan. She actually had an egg-sized tumor growing out of her forehead, and she had diffuse bone marrow infiltration. And you can see here that her tumor marker in the blood was going up. It had gone up higher than it had been prior to her second bone marrow transplant. And after a single intravenous dose of measles virus, that marker disappeared, the bone marrow normalized, the PET CT scanned resolved, and she had a durable complete remission, which has pretty much stayed under control since then. She had 2 small focal recurrences for which she had local radiotherapy, but she's now 7 years out off all therapy in complete remission. And if you just look at the next slide, which is a rendering of the CT scan that she had at the time of her therapy, you can see she had this egg-sized tumor on her forehead, which her family had called Evan. And you can see also there it was destroying the underlying bone. And if you go to the next slide, which is a PET CT scan, you can see that this tumor is bulging the skin, it's compressing the underlying brain, and before therapy, but in the repeat scan 6 weeks post-therapy, the contours of the skin and the brain have normalized, and that tumor is under control. Next slide. So the importance of Stacy was that she was a landmark for the field of oncolytic virotherapy. She was the first patient ever anywhere to get a systemically administered, intravenously administered oncolytic virus that targeted and eliminated disseminated cancer. And so she was special. And it's not been a walk in the park trying to replicate what happened with Stacy. Plenty other people have had the therapy, and the responses have not been so dramatic as they were with Stacy by any means. So why did she respond so well to the virus? Well, number one, she didn't have any anti-measles antibodies, so the virus could get at her tumor. And second, her tumor cells were very susceptible to measles infection, and they were immunogenic. So both the oncolytic and the immune phases of the therapy were pretty well taken care of. So next slide is just to talk about where we've gone since then. And because of this need to use a virus for which there is no pre-existing antibody in the blood, we moved to work with vesicular stomatitis virus, which is shown here. And this virus naturally infects cattle in Central America and the southern United States, and it causes a blistering illness that resolves fairly quickly. Farmers sometimes get infected and they get a self-limited flu-like illness. But the virus is a pretty powerful oncolytic, and so we turned our attention to this one because we could give it intravenously. Next slide. And this is just a little bit of data that was generated in our lab by Shruti Naik, who actually generated the virus when she was a PhD student in my lab. And what you can see on the right-hand side is that in this myeloma model that she's working with, if you treat the animals with a single intravenous dose, provided you give enough, you get complete tumor resolution, and those tumors do not grow back. So this virus is mediating both oncolytic and immune effects in this model. So we took it into the clinic. Next slide, please. And we actually first took it into the clinic in pet dogs because that was much, much faster than doing a long-winded dose escalation trial in people. And dogs do get a variety of different cancers. So we started working with this virus in pet dogs with lymphoma. And it was Amy LeBlanc who was at that time working at the University of Tennessee, who worked with us for this translation. And the next slide just shows that the virus was indeed active in dogs with T-cell lymphoma. And quite why T-cell lymphoma was the one disease where we saw interesting things in dogs, I'm not sure. But you can see here in a couple of dogs, there's a rapid shrinkage of involved lymph nodes at multiple different sites in these two dogs, which fairly rapidly regrows, but it nevertheless shows activity. And we believe that if we escalated the dose in this study, we'd have seen much more durable disease control, which is what we've seen in mice. So we moved the virus then into humans. Next slide. And we've actually treated a significant number of patients now at Mayo Clinic and elsewhere, both as investigator-initiated trials at Mayo and as company-sponsored trials by Vyriad elsewhere. And we've seen that when we give this virus intravenously, it's safe, it's not shed in urine or saliva, it inflames tumors, and it does have single-agent anti-tumor activity, especially in T-cell lymphoma and endometrial cancer. And it's also possible to track the spread of this virus because in its genome, it carries the interferon beta gene, which we engineered into the virus. So when cells are infected with the virus, they release interferon beta into the blood, and we can measure the levels to determine to what extent that virus is actually infecting and replicating in the tumor. Next slide. So this is just to emphasize that point. And here's a study that was led by Martha Lacey in hematology at Mayo, a phase 1 trial of this VSV virus given intravenously in patients with multiple myeloma and with T-cell lymphoma. And the interesting thing, if you look at the last column here, is that the interferon beta levels in some of the patients go very high, particularly here in patients 9 and 12. And both of those patients had T-cell lymphoma. And it looks, when we look at the responsiveness of the patients to the therapy, as if T-cell lymphoma is the best disease indication, because out of 5 patients with T-cell lymphoma, 2 had a partial response, meaning that their disease burden regressed by at least 50%. And 1 patient who was treated just in February this year has had a complete response with resolution of pretty extensive disease, particularly in the lower limbs. And this was really a nice thing for us to see another patient having a complete resolution of all their disease following intravenous administration of the virus. So, but the variability here is quite striking, and you can see from this, and I could show you a lot of other data that confirms this, that there are patients in whom nothing happens, where the interferon beta level is virtually undetectable. You can see that in patients 1, 6, and 7. There are other patients where there's an intermediate level of interferon, i.e. an intermediate level of virus amplification, and there are some where the virus really spreads quite dramatically. And so it would be very helpful if we could somehow identify at baseline those patients most likely to respond. So we started thinking about whether there might be a gene signature that would help us to identify responding patients. Next slide. And we thought very hard about what genes would be the most relevant to look at here. And we felt that the likely scenario was that tumors that were susceptible to virus infection were incapable of defending themselves against the virus. So they would have very low antiviral gene signatures. If we look at all the genes that protect cells against viruses, we expected those to be expressed at low levels, as well as those signaling and sensing genes that respond to viral infection and that increase the level of protection of a cell after it's been infected. And then finally, cells infected with viruses generally increase their expression of MHC and alert the immune system to the fact that they're infected. So we thought tumors that couldn't do that would be susceptible. So we came up eventually, after quite a lot of work, with a gene signature of 74 genes. And we have been looking at that gene signature in relation to the data we've been able to collect from patients enrolled in our clinical trials. And the next slide just gives you a sense of where we're at with that process at the moment. So here you can see the 74 genes. Blue means low expression, yellow means higher expression. And if you look down at the bottom, you have a series of green and red arrows. The green arrows are from patients who either had a response or who had stable disease. And the red arrows are from the patients who progressed. And it's not night and day here, but there is definitely a tendency for patients who have a cold tumor in regards to this gene signature being more responsive. So we feel we may have our foot in the door in respect of identifying patients most likely to respond. And in the next slide, I just wanted to show you that we went into the TCGA database where there are thousands upon thousands of data sets for RNA-seq assays done on tumors of various different types. And we converted our 74 gene signature into a Z-score. And so a negative Z-score here means that the tumor is relatively cold and should be susceptible. And a positive Z-score means it's relatively hot and should not be susceptible. And you can see that there's variability between different tumor types. In particular, you can see on the left panel that colorectal cancers do have a double hump, and it looks like they can be quite cold. And we have actually seen a fairly dramatic response in a patient with colorectal cancer. Pancreatic cancer looks like it's relatively hot and might be resistant. And then if you look across to the right panel, there's one tumor that jumps out there, which is uterine carcinosarcoma, which looks pretty cold. And we haven't yet had the opportunity to take a look at whether that tumor is particularly susceptible to our virotherapy. So I'm going to stop there. I've probably talked longer than was intended, but I think, it gives you a flavor. I think to conclude on what I've told you about, I think, it's clear that oncolytic viruses sometimes spread through tumors, killing the tumor cells and inflaming the microenvironment so that you get anti-tumor immunity, but sometimes they do nothing. And, more often than not, they do very little. Susceptibility or resistance to the virus infection may be predictable from the baseline gene expression pattern. And so we think that baseline gene signatures may be the key to success of this modality. But the alternative pathway that we're pursuing is to use drugs to combat the defenses that tumors do have in those tumors that are not susceptible to the virus. So the other approach we're taking is combination drug therapy. So I'll stop there and be happy to take any questions that anyone has, you know, whether about virotherapy, different viruses, about the sort of commercialization pathway that we've been pursuing, or whatever's of interest.
Thank you so much, Dr. Russell. Exciting and invigorating talk. Hopefully motivate a lot of us in the workforce to explore this avenue to help our patients. Perhaps I'm going to start with a question, and it goes back to the Stacy story. Were you surprised that the outcome was different for other myeloma patients compared to Stacy? And where is our current status in terms of management of our myeloma patients, and where do you see the future for patients with myeloma?
Okay, so with respect to the other patients other than Stacy, I was not surprised that we didn't get great responses. I was, however, disappointed because, you know, it would have been wonderful had we discovered the magic bullet and it would work every time. We rapidly realized that the majority of myeloma patients do have anti-measles antibodies and therefore wouldn't be a candidate for systemic administration of that virus. And we also determined that even when they didn't have anti-measles antibody, that didn't necessarily mean they were susceptible. So we're still working on that. You know, we've seen what can happen with Stacy, and I'm still constantly in contact with Stacy, who continuously encourages me to, you know, try and work out what was special about her and what we need to do in order to bring this to be a more generally applicable therapy. Patients with myeloma do have a lot of treatment options available. Unfortunately, the disease is still regarded as incurable, but with the fairly long list of currently available therapies, people can typically, you know, keep the disease at bay for something nearing now 8 or 9 years. And, you know, I think that's a great thing, but always it comes back, and so there's always a need for something new. We're seeing CAR T cells having an impact, but generally the responses after CAR T cell therapy are for a limited period of time. So, you know, 6 to 12 months is typically what people can expect from CAR T before the disease is back. There are new antibody therapies that are being developed. And it's certainly an area where there's a great deal of effort to develop a cocktail of drug therapies that will ultimately be curative.
Thank you, Dr. Russell. We've got a host of questions coming through, so I will start sharing them. One of the questions had to do with, where do you see virotherapy in 10 years, 20 years? And there was a comment about 2 patients who were reported to have died in a recent AAV clinical trial.
Okay, so those are 2 different things. So with the oncolytic virotherapy, I think it's going to be an important cornerstone of cancer therapy. And cancer therapy is evolving away from the use of chemotherapy, i.e. poisoning the tumor, to a new era where we'll be using the immune system to combat cancer. That's already begun with the checkpoint inhibitor antibodies and the CAR T cell therapy. Oncolytic viruses, I think, are a key component of what we'll be seeing in the future as you know, multimodality immunotherapy regimens for cancer. So I'm convinced that we're on a pathway to transform cancer care. In terms of the patients who died in the in the recent AAV study, these were children with a rare genetic disorder, myotubular myopathy, which was treated by intravenous administration of a high dose of an AAV vector that carried the gene that is damaged in these kids. Now, high-dose intravenous AAV therapy has been successful in other disorders. And so people have been becoming increasingly comfortable with the use of AAV vectors given intravenously. It's not 2, it's 3 children now who have died in that study because sometime post-virus administration, they developed liver failure, followed by multi-system failure. It's really a very worrying finding. It does mean that everybody's looking at their particular AAV vector. They're looking at the way in which it's manufactured, whether it contains impurities, what its surface looks like, and at the same time trying to understand exactly what went on with these children with this particular AAV vector. But it's not oncolytic virotherapy, it's gene therapy that's the therapeutic modality there.
Thank you, Dr. Russell. A few other questions. How do you establish tumor specificity of oncolytic viruses and avoid systemic disease in the recipient?
Okay, so in the early days of oncolytic virotherapy back in the 1950s, you know, when people responded to the virus, they were likely to then die as a consequence of the virus disseminating and causing brain damage or something else. Nowadays, we have much better technology for honing the specificity of the virus. One important point to make, though, is that viruses do have some kind of intrinsic ability to target tumors. Tumors lack lymphatics. Tumors generally hide from the immune system. And as such, they have no real strong defenses against virus infections. So they're intrinsically susceptible to viruses. So if you weaken a virus, if you attenuate a virus so it can't harm normal tissues, it will likely still be capable of damaging a tumor. But I call that soft targeting. What we really want for virotherapy is hard targeting, where we use antibodies displayed on the viruses so that they can only target cells displaying the appropriate target antigen, or that we incorporate into the virus's promoters so they can only be expressed in the tumor cells that we're interested in, or other mechanisms of hard targeting that we build in. And it's quite a big topic. There's a great deal of recent advance been made in that area. And I think the future is pretty promising for fully retargeted viruses that cannot infect and damage normal tissues being used for cancer.
Thank you. There are a few questions about side effects, and I believe you answered that. Another question had to do with protein signature. Is protein signature better than gene signature for viral treatment?
Don't know. Good question. And probably something we should look at. You know, one of the daunting things about diving into this area of gene signatures and, you know, potentially protein signatures is the cost and complexity associated with the generation and then bioinformatic analysis of this data that needs to be done. You know, I think we're very fortunate to be, you know, working inside Mayo Clinic on this, working with the people in the Center for Individualized Medicine to give us some firepower to look at it. But it, you know, it always has to be financed. I think with the proteomics on complex material that comes out of cancer cells at the moment isn't quite so clean as the analysis of RNA from tumor cells. But I think it nevertheless is worth looking into.
Thank you, Dr. Russell. Any relationship between responses to oncolytic viruses and the degree of tumor infiltrating cells present?
Hard to say at the moment, but based on the little amount of information we have, we think there is. We think that tumors devoid of infiltrating cells are more susceptible to virus-mediated destruction. On the other hand, though, we think that tumors that have a lot of infiltrating cells are more susceptible to the virus-induced immune destruction of the tumor. And so you're sort of between the devil and the deep blue sea there, that you have these two mechanisms of action which do connect to each other, which mean that you could potentially have a response even if you do have a significant immune cell infiltrate in the tumor.
All right, the questions keep coming. What are the top oncolytic viruses used currently?
The only oncolytic virus that has gained FDA approval here in the US is called TVEC. It's manufactured and distributed by Amgen, and it's a recombinant herpes virus, herpes simplex type 1. It's been engineered to remove a couple of the genes that it uses for pathogenicity. So this is an attenuated virus. And it's been engineered to encode a cytokine, granulocyte macrophage CSF, GM-CSF, and it's injected directly into the tumors that are being treated. It can't be given intravenously. And it's been approved for treatment of malignant melanoma. And if you inject the virus directly into melanoma tumor deposits, you often see that uninjected tumors will regress, as well as the one that's been injected with the virus. So herpes simplex virus is now the lead oncolytic virotherapy platform in terms of clinical use. There are numerous programs developing new and better versions of herpes simplex virus. There's a recent study been completed in Japan to show that patients with glioma have significantly prolonged survival when they receive repeat injections of this, a different herpes simplex virus into the brain tumors that they have. So instead of having brain surgery. So herpes is an important one. Adenoviruses are being developed. There's a picornavirus, Coxsackie virus A21. There's a Rio virus, and several other viruses in the pipeline. Vaccinia is also quite popular. But there's quite a menagerie of different viruses being developed. Our own interest, research interest at Mayo, is for intravenous therapy. And that really causes us to confine our attention to viruses now that don't need to be given intratumorally and which can be given intravenously. Because we really want to reach all of the metastatic tumor deposits when we deliver our viruses.
What other tumors have you treated with this approach, and how is your success or lack of it has been?
So, as I mentioned, T-cell lymphoma, the endometrial cancer study that's going on at Mayo, led by Dr. Bakam Gamez, is looking very interesting, and there have been patients in that study who have had significant benefit from the virus administration. We, as I mentioned earlier, have had some experience in colorectal cancer and have seen activity there. Viriad as a company has just launched a phase 2 clinical trial in collaboration with Regeneron Pharmaceuticals. And the trial design is to combine an intravenous infusion of this VSV virus with Regeneron's cemiplimab anti-PD-1 antibody. So there's a great deal of interest and excitement at the moment in the whole virotherapy world of combining checkpoint antibody therapy with virotherapy. We have quite a lot of preclinical data says that that combination is good. And so that trial's moving forward in 4 indications initially: melanoma, lung cancer, non-small cell lung cancer, hepatocellular carcinoma, and endometrial cancer. And that study just opened on July 15, so it's accruing in multiple centers as we speak.
Great. There are a couple of questions relating to interferon beta. One has to do with the concern that extended dosing would be difficult with a single vector. Do you have a set of viruses expressing interferon beta to enable an extended dosing approach? And the other one had to do with VSV interferon beta replicating in patient tumor. Is there evidence for that?
Yeah, and the evidence for VSV replicating in patients' tumors comes primarily from looking at that interferon beta profile, because in clinical settings, it's very difficult, and in a phase 1 clinical trial, it's very difficult to justify sticking a needle repeatedly into a patient's tumor. That was done in one of the clinical trials we have, and we have definite evidence that the virus does and can amplify inside the tumor, and that amplification correlates to the interferon beta levels that we see in the bloodstream, and the sort of area under that curve tells you how much and how far and how long the virus has been propagating. So the interferon beta is a really good reporter gene to tell you what's going on with the infection, because without that you just don't know whether the virus is amplifying or not unless you're doing repeat tumor biopsies. So that's on that question. On the other part of the question as to a sort of panel of different viruses encoding interferon beta, I think that's a wonderful concept. And the idea of sequential virotherapy using different viruses for a single patient with a single type of tumor would be very appealing, because we know that different viruses may have a different probability of being successful in a given patient, and that the identifying which virus is going to work for a given patient is difficult. And by giving a series of different viruses, you sort of overcome that. Problem is, each new virus that you give is a new product, requiring quite a lot of expensive process to take it through the manufacturing and clinical testing required to have it incorporated as an approved agent in that kind of protocol.
And our last question for this webinar is are clinical trials required to get virotherapy approved by the FDA?
Absolutely. FDA is, I think, it's the organization that determines what is allowable, what data has to be provided in order for any kind of clinical testing to go on. it is responsible for determining what kind of data is needed before final product approval. And, you know, it looks at every aspect. You know, I think in terms of final product approval, what the FDA wants to know is that the virus that has been developed is, number one, safe. Number two, that it is an advance in terms of efficacy over the best standard of care currently available. And so, you know, in the longer run, it is critical that phase 3 trials are conducted, pivotal phase 3 trials, in which the virus as therapy, either alone or in combination with other drugs, is compared with the standard of care in the cancer in question. And the virus has to show a superiority of some kind, whether that be a better survival, a better response rate, or an equivalent survival / response rate with lower toxicity. It's got to meet that kind of requirement. And then FDA looks at the manufacturing, and they will not approve a drug unless they are quite confident that each batch of drug that is produced is going to perform exactly as advertised. So the manufacturing process has to be really very well characterized, and batch to batch variability has to be no longer an issue. So yeah, very rigorous, very long term, and huge amounts of money therefore required in order to get through the process, and a long time is required to get the FDA approval.
Thank you, Dr. Russell. Any lasting thoughts or comments to inspire our audience before we end?
Well, what I would say is that, you know, engineering viruses is quite addictive. They're rather simple in that, you know, even the large ones have a limited number of genes. The function of those genes is fairly well understood. The life cycles are fairly well understood. And you can make changes to the viral genome by introducing new genes in or adapting the genes that exist to make the viruses behave in a way that makes them more suitable for cancer therapy. And it is the world's best Lego set, and it's a lot of fun generating new viruses and determining what they do. So, you know, anyone who's thinking of virus engineering, virotherapy as a career, I couldn't recommend it more strongly. And I really think we're at a point now where we are beginning to have that critical impact on cancer.
Wonderful. Well, thank you everyone for joining us. Please watch for a follow-up evaluation that will be coming soon, and mark your calendar for September 11th from noon to 1 for the Exposomics 2020 and Beyond webinar. We're also hosting a virtual Individualized Medicine conference on October 14th for $75. You can join us online for 4 hours and learn more about individualized medicine for your patients. Thank you all for joining us. Bye-bye.
All right, thanks very much, and thanks for all the questions. Okay.
Thank you, Dr. Russell.
Bye.
Bye-bye.